Hi, I'm Elsa Thompson with Survey of Chemistry, and this is my chemistry video number 4 for section 4, chapters 10 through 12. Just gonna jump right in. This is number 1. So, to start, this one is about proteins. Answer the following about proteins. The question A says, proteins are polymers made of amino acids. Draw the structure of the dipeptide THR, LEU, and label the peptide bond N-terminus and C-terminus. So here it is right here. Here is THR, LEU. And this is mainly the THR part. And then this is mainly the LEU part. And this is the N-terminus because it has the H3M plus on that side. And then this is the C-terminus. I don't really know, because it has the O minus. So, and this is the peptide bond, more specifically, here is the bond, because it puts these together like that. So that's number 1. Or, I guess, A. B is protein structure can be classified as primary, secondary, tertiary, or quaternary. Explain what types of forces stabilize these structural levels. In your explanation, be sure to point out how each of these structural levels are different. So here's one. One is held together by peptide bonds, and it is like a backbone. It's like a chain, long chain, like a necklace with all like the different amino acids on the little, here's our necklace, and here's our little amino acids. Okay. Our little, these guys. Okay, so then 2 degrees, secondary, is held together by hydrogen bonding. There's 2 kinds of secondaries. There's an A helix, which is like bending the backbone, so it's just like, oh gosh, it's gonna be a terrible drawing. Yeah, not very good. Like, okay, whatever. There's a B pleated sheet, which is like when you fold a paper like back and forth, it's just like a pleated sheet. Like, I don't know, bad drawing. I'm a bad drawer. Anyways, yes. So then, tertiaries held together by London forces, hydrogen bonding, ion dipole bonding, dipole, ionic salt bridges, and disulfide bonds. Which is, these tertiary ones are basically when secondaries react to bend and shape itself into like instead of having those, my terrible drawings, it's like all like wacky with like different salt bridges, and there's a salt bridge, there's some hydrogen bonding, I don't know, just like that. Okay, and then quaternary has the same forces as tertiary, so the London hydrogen bonding, ion dipole, ionic bridges, and disulfide bonds, but it's between subunits, and it's when the tertiaries, we have 2 or more tertiaries that like link together, so you get a bunch of the little snake buddies that all go together and make a big glob. So they basically just all build on each other. So yes, okay. My C's up here. C says enzymes are protein catalysts that increase reaction rates by lowering the activation energy. Describe 2 types of enzyme substrate interactions that lower activation energy. You may wish to include drawings in your explanations. Okay, my first one is, let me zoom in. Okay, is proximity, which is the closer the enzyme, closer to the enzyme active site, which would be like right here on this picture. And the reacting molecules are the more likely and the more easily react, this lowers the activation energy. So I put this one as like a check mark because the let me put the cap on this pen. Because the here's our little reacting molecules, because they're going into the enzyme and they can activate more easily. So you have lower activation energy because it doesn't have to try as hard. And then if it's way out here, that's bad because it's probably not going to react. Okay, number 2 is orientation. If the reacting molecules are not positioned correctly, the substrates will just bump into each other, so they might not react. So here's our 2 things that need to react with each other. and it lowers activation energy. The enzyme helps because they get positioned into their correct little spots in the enzyme, just right here. So then this part of it can go into that part, like a little puzzle. Okay, so that was my number 1. Moving on to number 2. Number 2 talks about nucleic acids. I have lots of writing. Eek. Okay, so A says draw the structure of a nucleotide. In your drawing, label the phosphate, sugar, and base. So here's my nucleotide. This is CMP. Here is my phosphate right here with the P and bonded all the oxygens. And then here is the sugar, the 5-carbon sugar. 1, 2, 3, 4, 5. And then here's my base, which is I did a pyramide, I don't know, something like that. But it's one with just one. So there it is. My B right here. It says contrast DNA and RNA by explaining how their molecular structures are different. Then explain how their functions in the body are different. So DNA is a double helix with anti-parallel backbones. Contains junamine, I don't know how to pronounce these. Cysteine, adenine, and thymine. So A goes to T and G goes to C. RNA is just single-stranded. It copies a gene from the DNA. Contains the same things up here as DNA, except it has uracil instead of thymine, so it goes A to U. The functions are that, to put it simply, DNA makes, it's just like, so one strand of the DNA is the same as the RNA because it copies it, and the RNA makes proteins with the help of other things in the cell. But so the DNA holds the information while the RNA copies it and uses it to make proteins and stuff. Okay, so my C, it first wants to know, so its viruses are small particles containing genetic material that can infect any cell type. Describe the structure of viruses and explain how viruses infect the cells and replicate. In your explanation, be sure to use the following terms: host cell, nucleic acid, capsid, envelope, protein spikes, and budding. Okay, so this is how a virus is structured. Let me zoom in. So a virus has a shell with protein spikes on the outside, the envelope, which is on the outer layer, the capsid, which is a part of the shell, like the inside part. It's right there. I should get to that later, but it's made out of protein, and the virus is filled with nucleic acid. So here you can see this color. This is the nucleic acid. It's this stuff in here, and then here's the little capsid. They're like little balls in there. And then the envelope, I kind of tried to draw it 3D. Kind of bad at it, but it's that part on the outside. And the protein spikes are these. Oh, guys, there's more protein spikes and stuff, but I can't fit them all in there. I have terrible drawing. Okay, a lot of words, but I would read them off. So this is how a virus infects a cell and replicates it. Okay, an enzyme on the protein's outside will make a hole in the host cell. So here's the outside of our virus. It makes a hole in our cell. Oh no, hole. Terrible drawing. And then the host cell is the infected cell. And then the virus's nucleic acid will mix with the host cell's insides, just like its materials. The host cell will then replicate the viral DNA inside the nucleic acid, and then make proteins to make more viruses. The new viruses will be pinched off through the outside of the cell. So they'll go in here through the cell, and they'll be pinched off on the outside through a process called budding. The new viruses will then go wherever they please and infect new cells to spread. So that concludes 2. Moving on to C. Lots of words. It'll be okay, I promise. I know you're so worried. Okay. Oh my god. Okay, this one is about metabolism. Okay, so the first question is define catabolism and anabolism. Then name 2 types of chemical reactions that tend to be found in catabolic pathways, and then 2 types that are in anabolic pathways. Okay. So catabolism is when larger molecules are broken down into smaller ones, and you have hydrolysis and oxidation. Anabolism is when smaller molecules are reacted to make larger ones, condensation and reduction. So basically they are the opposites of each other, because this one is large to small, and this one is small to large, and this one has hydrolysis, this one has condensation, this one has oxidation, this one has reduction. So they're just like interchanging opposites of each other, like larger to smaller, smaller to larger. was moving it all over the place. Yeah, they're the opposites of each other. They all happen with metabolism, so. All right, then B says, oh gosh, endogenine triphosphate ATP, called the energy currency of the cell, is hydrolyzed to diphosphate ADP in various cellular processes. Draw a chemical equation showing this reaction, then label ATP and ADP as comparatively higher or lower in energy and explain whether the reaction is exergonic or endergonic. Okay, so here is my chemical reaction or chemical equation. So we have our ATP plus H2O goes to ADP and PI, and then free energy. Energy is released. So that makes this exergonic because it's being released. ATP is high energy and ADP is low energy, because you can see right here it has triphosphate, so it has more, so it's higher. And this one has diphosphate, so it's lower. And then here's our endogenine, which is the A, and then this is triphosphate, so it's ATP. Here's our A, so it's A, and then diphosphate 2, diphosphate DP. So there's that one. All right, a lot of words. Don't worry, I will read them off and we'll get this done. Digestion is the process in which large fuel biomolecules are broken down into smaller hydrolysis products. Goodness gracious. These hydrolysis products can then be absorbed by the body and delivered to cells. Explain how starchy meal made of polysaccharides is digested in the body. Be sure to describe how enzymes are involved in the mouth and small intestine. Okay. I basically went through the whole entire thing. So here we go. We have our polysaccharides go from the digestive system, which is like part of the mouth and small intestine, into the cell's cytoplasm, and finally to the cell's mitochondria. So first in the mouth, the polysaccharides are broken down through the enzyme called alpha amylase, and further broken down in the stomach, the small intestine, with the enzyme called pancreatic amylase. Okay, so that's the first part. So this turns it into monosaccharides. So then next at the cytoplasm, the polysaccharides have been previously made into monosaccharides right here. So those turn into pyruvate through a process called glycolysis. I think I'm saying that right. Glycolysis, I don't know. Glycolysis is regulated by many different enzymes and the presence of ATP. Okay, so now we have pyruvate past here. Under anabolic conditions, which we talked about up there. Anyways, through anabolic conditions, the pyruvate is made into acetyl-CoA. I don't know what to say that. CoA through oxidative decarbonation with help of the coenzyme A. Okay, so now we have that CoA. Next, the CoA is used in the citric acid cycle, which makes a lot of things, but to highlight a couple, we have 3-NADH and FADH2. So now we have those two things. Those are put under the electron transport and oxidative phosphorylation to make ATP and H2O, and also the NADH turns into NAD+, and the FADH2 turns into FAD. So now we have, finally, ATP and H2O. So this is what's made at the end of the whole entire everything. So that's what happened when I ate my crackers today for breakfast. I didn't even know what was happening. I do, I guess. I do know. But anyways, okay. That concludes my video. So thanks for watching. And I hope you enjoyed the video. I'm saying this like it's a YouTube outro. Is it A-worthy? I guess I'll find out. Okay, anyways. Thanks. Bye.